Greetings to you all and welcome from the New York Botanical Garden. I'm Lawrence Kelly, Vice President for Science Administration and Director of Graduate Studies, and I'm happy to welcome you to the symposium, Here Today, Gone Forever, Plant Extinction Now and Conservation Strategies for Tomorrow. Our purpose is to shed light on the problem of plant extinction and also to explore strategies to minimize future extinctions. We owe huge gratitude to our speakers today, plant experts and biodiversity specialists from far and wide who are here to share their perspectives and their data to shine a spotlight on the topic of plant extinction. Our speakers affirm the values of the New York Botanical Garden, specifically the essential role of plants in ecosystems and the health of the planet, the necessity of plants for our survival, and the importance of data-driven science to understand and conserve plant diversity. Before going any further, I would like to inform you that this symposium is being recorded and saved by the New York Botanical Garden, and it will be available for viewing in our online lecture library. It is my pleasure to introduce our moderator for today's symposium, my colleague, Dr. Robert Nazi of the New York Botanical Garden. Rob?
Thank you, Larry, and greetings everyone from the New York Botanical Garden. I'm Rob Nazi, the Arthur Cronquist Curator of North American Botany here at NYBG. I'm very excited to join Larry and welcome you to this symposium. Shortly, we will proceed with pre-recorded presentations from today's 4 experts. Immediately following the presentations, we will have a live question and answer session with the speakers. Feel free to send questions during their presentations. At the lead of your question, please mention to whom you direct it. If you are using Zoom, please use the Q&A button. Also, if you have the same question as someone else, please give it a thumbs up so we know to prioritize the most popular questions. You are also welcome to use Zoom's chat feature to comment, but please use the Q&A for your questions. If you are on YouTube, please use the chat feature to send your questions. Presenters for today and next week bring diverse backgrounds, perspectives, and styles. In order to provide as much time as possible for the presenters and ensuing discussion, I will keep my introductions brief and invite you to view the speaker's biographical information on the symposium website. At this time, I am pleased to introduce Dr. Thomas Lovejoy, who will introduce the symposium. Tom is University Professor of Environmental Science and Policy at George Mason University.
So good morning. Welcome to the New York Botanical Garden's 2-part symposium on plant extinction and conservation. Here today, gone forever, plant extinction now and conservation strategies for tomorrow. A great 2-day program organized by Rob Nazi and Larry Kelly of the New York Botanical Garden. I'm Tom Lovejoy, a trustee of the New York Botanical Garden, and I couldn't be more delighted to welcome you to this 2-part effort to kick off a major effort on the part of the New York Botanical Garden to engage with the larger plant conservation community and create a much rosier future for the variety of plant life with which we share this planet. So I want to begin with a set of images. These are the images that the garden used to essentially highlight the announcement of the symposium. And so it starts with some U.S. species that are endangered or threatened, the flowering mountain pitcher plant, listed as threatened by the US here in greater number. The fruits of Short's goldenrod, also listed as endangered. The western prairie fringed orchid, listed as threatened by the US. The swamp pink, also listed as threatened by the US. And finally, a sober reminder of what can happen if we don't act. This is the intertidal marsh habitat where the extinct Nuttall's mudflower once grew. So let's get back to thinking about a little bit about the challenge before us and the opportunities embedded in the next couple days. So I thought I'd start with those images just to remind us of what many of us refer to as plant blindness. The tendency of most people to look at images of nature and see the animals, but not the plants, which are an integral part of their habitat and ecology. So hopefully part of what we can do in the activities spinning out from this symposium and future collaborations is actually get people to open their eyes to the plants, which basically support the animals that they have been focusing on in a kind of ecological myopia. So I don't need to elaborate the scale of the problem of plant extinction or extinction generally. Habitat destruction is still probably the greatest single factor, but climate change is thundering up on the outside. And I think it's important to realize that basically anything we call an environmental problem ends up affecting biological diversity because they're only called environmental problems because they affect biological systems. So biodiversity integrates all those negative things, which makes it the single most difficult environmental challenge to take on. That said, I think we are positioned to make a much greater difference going forward. And using modern scientific tools not available until very recently, such as artificial intelligence, the use of all the stored digital images of herbarium specimens, and the newest in remote sensing imagery, like Dynamic Earth, which will be providing 10-meter resolution, updated annually by artificial intelligence, totally free for non-commercial purposes. So there's a lot of opportunity embedded in this symposium, and I look forward to learning with all of you about how we can make a much bigger difference. Thank you very much.
Thank you, Tom. The next speaker is Dr. Stuart Pym, the Doris Duke Professor of Conservation at Duke University. Stuart will present Preventing Future Plant Extinctions.
Thank you so much for giving me the opportunity to speak to you about plant extinctions. I know that we have a fantastic slate of speakers, many of whom are going to talk about related issues to the ones that I'm going to cover. What we know about the current extinction crisis is rather neatly summarized by Al Gore in An Inconvenient Truth. He talks about species going extinct 1,000 times faster than normal. If you wonder from whom he got that, the answer is from me. I'm even more flattered by this demonstrator in England holding up a sign saying extinction rate up 1,000-fold. My work has never been featured in a street demonstration before. I am so proud. The number of 1,000 times is worth exploring because I think it's relevant to understanding of both the nature of where we are and what we need to do to prevent future extinctions. The obvious question is, why did Gore say it in this way? Why did he talk about the extinction rate? What's simply talking about the numbers of species going extinct per day? Well, the problem with that is we really don't know how many species there are. As we'll see, we have a pretty good idea for plants, but apart from a few vertebrate groups, we don't know for the great majority of species, particularly insects and fungi. So we have to use some other metric. So I want to address 4 questions. First of all, I want to talk about how fast evolution is creating species. So we have a reference measure. Then I want to talk about how fast species are going extinct. What are we doing to protect them, and how might we do better in the future? Well, it's a long story to calculate a background rate. And I was fortunate to work with Yuri de Vos on some of the more complicated statistical aspects of this. But in brief, fossil death rates are really hard to interpret. We can get some interesting data out of molecular phylogenies, and by far the simplest measure is just simply to talk about how fast evolution is creating new species, as it were the birth rates of species, and to compare those with what we're doing now. This is an example of a molecular phylogeny. It's a rather complete one from South Africa for an orchid genus. And if you look at the rate at which species are being formed according to this molecular phylogeny, you see that birth rate is approximately 1/4 of a new species per million species per year. So for each million species, you'll get 1/4 of a species each year, 1 new species in 4 years. And that number is actually quite high relative to other phylogenies, but it's broadly in the same order of magnitude. Species are being created at that level. So how fast are species going extinct? Well, for that, we can talk about the death rate. We've talked about the birth rate, now let's talk about the death rate. By analogy to humans, we know what our death rates are. A background death rate for the world is about 8 people per 1,000. Obviously, at the moment, that's substantially higher because of COVID. So we know what the normal rate is, and we express that rate as, deaths per 1,000 people per year. Under wartime conditions, the rate goes higher. So we have a sense of a comparison to normal conditions and appalling conditions during conflict. Extinction is a death rate too. We can talk about extinctions per million species per year. We express it that way so that the numbers are, sensible ones. And when we do that, we know for birds and amphibians and mammals, and recently for plants, and we're going to hear about that in another talk. If we look at those numbers overall, we see that they're measured, roughly on a scale for vertebrates, at least, of 500 to 5 50 to 500 extinctions per million species per year. We can follow a cohort of species from the time they were described to the present and look at how many of them have become extinct in that period. But if we look at these data quite consistently, what we see is that the species that have been described more recently have higher extinction rates than those that were described, say, in the 19th century. That's true of birds, it's true of amphibians, that's true of mammals, it's true of plants. The reason for that is important to my story. But the first point is that the basic comparison is that we know that we are driving species to extinctions about 1,000 times faster than species diversify as judged from molecular phylogenies. It's a complicated story to relate those to the fossil record, and I will skip that. But, and it's a big but, recently discovered species have higher extinction rates. And that's certainly because they're rare. So how many species are there? And how many species remain unknown? Now, we have names for 390,000 vascular plants once adjustments have been made for synonymy. Can we estimate how many species remain unknown? Well, the basic idea comes along these lines. Let us look at the number of unicorn plants. Yeah, I know, that's a bit of a cheat, isn't it? And let's look at how many of those species are described within herbaria. And yes, I do understand that that is not the New York Botanic Garden, but I owe eternal fealty to Peter Raven. And so obviously I have to honor him and his garden. The basic idea is this. Supposing we suddenly come across a new part of the world and we realize there are all sorts of undescribed species of proboscidea out there. At the moment, there are none in herbaria. In the first decade, we might describe a bunch of them. In the second decade, we'll describe fewer. The third decade, fewer still. The fourth decade, you see the idea that the more known species there are, the harder is it going to be to find new species. So this collector's curve, the fact it gets harder to find new species the more you have found, has a simple model. It's an exponential decrease in the number of species being described, and with a little bit of simple mathematics, we should be able to predict how many more species there are to be found. The only problem with this is it fails badly. It fails really spectacularly poorly for all sorts of groups, especially plants. So if we look at the number of species, in this case of non-monocot plants, this is a sample, incidentally, of taxonomically revised species. There is no clear tendency for the numbers of species described to be dropping off. The only reason there's a decline in the last 5 years of this study is almost certainly that the descriptions have not been properly catalogued. And indeed, from about 1950 to the present, the number of species described has been going up, not down. The cause, of course, is that there are more and more taxonomists. In other words, the effort to find and describe new species is increasing. What we can do is to look at the ratio. We can look at the number of species described per taxonomist, and it's dropping. It's declining and has been declining for over a century. And very boldly, we can extrapolate, and we can say that by 2030, a plant taxonomy will end, and we'll have described 15% more species. Do I think plant taxonomy will end in a decade? No. but it does likely suggest that overall that we have, we are exhausting the supply of new species. It's taking more and more taxonomists to find fewer and fewer species. So, with about 15% more species to be found, recognizing that they're likely to be species with small ranges and they're likely to be rare, which is why we haven't found them, we should be asking where those species are. Are they in danger? Are they in a place where they are likely to go extinct? Indeed, are they in a place where we might not find them before they go extinct? Well, Norman Myers, late Norman Myers, came up with the idea of biodiversity hotspots. The idea that there are concentrations of species with small geographical ranges. And moreover, Norman realized that these are the places where habitat destruction is most extensive. Since we know that plant and animal species described since 1900 have 2 to 3 times higher extinction rates than those described earlier, it's likely that they might well be in these places where the extinction rates are going to be even higher still because of the destruction. And so we can begin to look at those models region by region by region. And when my colleagues and I, including Norman, did this, we recognized that the biodiversity hotspots, the places like Ecuador, are special in that when we look at the numbers of species being described relative to the numbers of taxonomists, that trend line is remarkably flat. There is absolutely no tendency for the number of species described per taxonomist to be dropping in Ecuador. It's pretty much the same rate for the last 200 years. That means that we can only anticipate that there's going to be a lot more species described there. In short, it's the biodiversity hotspots that house most undiscovered species of plant. We know those places are under massive threats from deforestation, and consequently, those 15% of plant species which we anticipate may be unknown are living in places where they are at great risk. In other words, the extinction rates are going to go higher. So finally, how well are we doing at protecting biodiversity? Now, this is a subject close to my heart, and I wish I could ramble on for hours on this subject, but I can talk about one important piece of the puzzle. Let's look at where we have set aside our protected areas worldwide. We've protected a little shy of 18 million square kilometers of the terrestrial land surface. If we look at where those places are, they are typically in wilderness. By wilderness, I mean places which have the lowest human impact. And I particularly wanted to look at the half of the Earth, for reasons I'll come back to, the half of Earth that has the lowest human impact. And our protected areas are predominantly in those places. Only a small fraction of protected areas are in the half of Earth that has the greatest human impact. The reason why I'm interested in this problem is that next year in Kunming, COP15 meets to plan out the priority for the next decade of biodiversity protection. The Convention on Biological Diversity's 15th meeting is going to come up with an agenda that says, what should we do next? And if we simply talk about area, Do we want more area? It's likely that we're going to get what we've got already, which is large areas with very few people in them. So it's reasonable to ask whether there are a lot of species in these wild areas. And if we just simply grab area for protection, will we protect more wilderness and will we get more species as a consequence? I mean, an example of this is, I believe, the world's largest national park. It's at the top end of Greenland. The only reason I've been there, why would a tropical biologist like me be in northern Greenland, is it's on the plane route from Shanghai to the US. And so I fly over it when I go to China. And that was a clear day when I took it. I have no idea what the plant list is for that national park, but I suspect you can probably count the species on the fingers of one hand. Protecting wilderness areas does not particularly protect biodiversity. What we can do is to look at how well we have protected biodiversity as a consequence of what we would expect under the null hypothesis. And the argument goes like this: supposing we have a large range of species, you'd expect 13% of it to be in protected areas, half in wilderness, half in not wilderness, arranged in this Venn diagram. You might say, well, what happens to our unicorn plants, which have small geographical ranges? Over the entire ensemble, you'd expect 13% of them to be in protected areas, some to be in wilderness, some to be not. So we can do this with, okay, not plants, but the vertebrates that we know, birds, mammals, and amphibians. And the remarkable thing is that when we look at species with small geographical ranges, the most vulnerable species, we have allocated our protected areas really rather well. We've done a good job, much better than chance, of protecting species with small geographical ranges than the allocation of 13% of the Earth's surface to protection would expect. I'm pretty sure that's going to be true of plants. That by picking the biodiversity hotspots and creating relatively small protected areas, we have done a better job of protecting the most vulnerable species. But the question is, can we do better? And that brings up the agenda for COP15 in Kunming next year. Well, there are a variety of initiatives. One of them is Ed Wilson's Half Earth. And Ed has said, look, we need to be protecting half of Earth. That should be the target. The problem I worry about that, as you've seen from the previous slides, is that just by protecting big chunks of land, we are not necessarily going to protect more biodiversity. Protecting more of Greenland, protecting more of the Sahara, protecting large chunks of sparsely populated China, and maybe politically easy targets, but they're not going to do a lot for biodiversity, plant or animal. We need to be thinking strategically of what the target should be. The official Chinese position now is to talk about a target of 30%. You know, 30% is not 50%, it's a great target, but unless it's appropriate, we're not going to capture a lot of species. So, you know, do Ed and I disagree? Well, sort of. What Ed has agreed is that we need to be smart in what we ask for. Half Earth represents a powerful conception, but we need to think about what those priorities should be. I think the challenge is to come together internationally with an agenda that will protect biodiversity, not just merely fall prey to simplistic targets of area. Thank you so much, and I look forward to taking questions.
Thank you, Stuart. Remember, please feel free to send questions during the presentations using the Q&A of Zoom and also the chat of YouTube. I appreciate the questions that are coming in, but feel free to send more. And please do indicate the presenter to whom you direct your question. Our next speaker is Dr. Maria Vorontsova, Research Leader for Grasses at Royal Botanic Gardens, Kew. Maria will present Modern Plant Extinction, What We Know.
My name is Maria Vorontsova. I'm a taxonomist at Kew Gardens, and I will briefly take you through my journey with plant extinction. I first came across plant extinction during my postdoctoral work on the spiny solanums of Madagascar. And I found myself in this coastal Tanzanian area called the Ruvu Forest, searching for the plant you see here in the picture, the spiny solanum I came to name as Solanum ruvu. And we rapidly discovered, and local people helped us understand, that Ruvu Forest had in fact become this agricultural landscape with rice paddies that you see here, banana trees, and occasional little wild thickets not capable of supporting large trees. And to cut a long story short, we never found this plant. And many years later, I was in the middle of Madagascar here, searching for the unique endemic Sartidia perrieriae that you see in the picture. And we searched everywhere for a number of years, and Sartidia perrieriae was nowhere to be seen. I felt that we needed to communicate the fact that these plants were missing to the global audience. And so I published preliminary conservation assessments for both of these species during the routine course of my work. And I came to understand that, in fact, my situation was really very common. So many unique tropical plants, especially tropical, are known from just one collection, one type specimen. And often these locations are not revisited and these plants are not found for many years afterwards. But I realized that the majority of my colleagues make a different choice from me. And when plants are missing, nobody publishes about it, because proving extinction is actually impossible. How do you prove that a plant is absent from a really big, complicated landscape, and that it's absent from the seed bank and it doesn't come back every 10 years? Proving things is difficult. So there are other extinct plants I'm sure you've come across that do have a big glamorous profile that people know about. For example, this rather expensive Encephalitis woodii that I definitely couldn't afford to have in my garden. My journey with extinction also starts with my Kew colleague called Raphael Govertz. This is Raphael in his back garden, indulging in his hobby of understanding extinct plants and growing extinct plants and looking for them. Before coming to Kew, Raphael was looking for a list of plants that had gone extinct. And back in the 1980s, he realized that actually nobody had ever made a global list of extinct plants. So after he came to Kew, he scanned all botanical literature we have available in our library, grey literature, reports, floras, checklists, to make a big database of everything published on extinct plants. And the results of this analysis is what formed the backbone of the paper we published last year. So Raphael's global literature compilation identified an awful lot of names that we had to reconcile taxonomically on a global level. And we concluded that 1,319 species of seed plants had been published as extinct. So nearly 600 extinct, or extinct in the wild, still alive in botanical gardens and collections, and a further 431 rediscovered. The first question we asked during our research was, why do some plants go extinct and others not? And we expected to see an evolutionary pattern where maybe some lineages are weaker than others. But our formal analysis of the seed plant phylogeny seemed to show that extinction so far, our records of extinction, are phylogenetically random. But instead of having an evolutionary driver, there seems to be some kind of driver linked to geography. So throughout this talk, I'll be showing different ways of mapping the same data set, the same big data set that we've been working on. This is the first one of those maps, and this shows simply the number. How many seed plants are extinct fully or extinct in the wild, not rediscovered, in which countries. This mapping scheme that you see is countries or first level provinces for big level countries. And it's a Tadwig scheme originally put together by Dig Brummett Q. Unfortunately, a bit incompatible with animal data, but that's the scheme that many Q data sets are based on. So the first thing about this map that's pretty important is all the white spaces. And every white space on this map is a country where no extinction has been recorded. Nothing is extinct. Then we ran a second analysis looking at what factors are particularly associated with extinction. And this cyanea, very beautiful from Hawaii, represents the biggest risks that we've identified. So unsurprisingly, the most extinctions have been recorded from islands, from the wet tropics, and woody perennials appear to be the most at risk. So here is the second part of our data set, and that's only plants that have been declared extinct and then they have been rediscovered. And some of my colleagues seem to think that the fact that rediscovery happens somehow means that extinction science is bad quality, or it somehow discredits the original declaration of extinction. And I'd like to challenge that viewpoint here. My suggestion is that when we suspect something may have gone missing, we need to raise the alarm as soon as possible. And it's quite natural that the process of knowledge building should lead to things being missing and then things being found again, and then maybe them being missing again. There's nothing wrong with that. You'll see the rediscovery pattern. It's a little bit different. There's still an awful lot of white spaces, though, all of those white spaces where nothing has been extinct and then rediscovered. This is one example of a plant you may be familiar with that has been rediscovered, Tecophyllaea cyanocrocus. I unexpectedly found myself on the TV rather a lot last year to present the results of our extinction paper. And after many years working in this area, the media exposure really made me realize that the word extinction can help draw the public's attention to plants, to botany, to ecology, to all this deep and complicated work that we do to study our ecosystems. And ordinarily, plant blindness stops people really from paying much attention. But since extinction has such a high profile, that could be one way for us to connect our knowledge bases with the audience that we want to reach. I now reach the second part of my talk, which is the work we did this year to understand the inequality of botanical knowledge and dig into why exactly is the extinction pattern so uneven across the world. So now you see the third map of exactly the same data set of what has gone extinct. The islands in red have the greatest proportion of their total seed plant diversity extinct. And again, all of those pale green areas where nothing has been declared extinct, I want to show you this fantastic analysis by Carson Mayer, published in 2015, which shows that for vertebrates, at least, the richer the country is, the better their coverage of vertebrate species records. Let's look at Africa, which is in pink and purple. It has the lowest wealth overall in the world, and we can see that it has clearly the lowest vertebrate species inventory completeness. So does wealth and GDP have anything to do with it? This is my fourth and final global map of the same data set, and this time pink areas are where extinctions have been declared but things haven't been rediscovered. Green is where extinctions have been declared and everything has been rediscovered again. Dark gray are places with multiple extinctions and multiple rediscoveries. So I'd like to suggest that the dark places have had the most botanical work and the most effort, since they have species at all stages of the process and they have accumulated significant data sets. So we can see significant data sets on plant extinction. They're in Australia, they're in India, they're in parts of China, they're in South Africa, southern United States, Mexico. The greatest amount of work in recording extinction and rediscovery, all of it has happened in countries that are not necessarily particularly wealthy, but countries that made a good investment in recording and tracking their biodiversity, countries with a healthy botanical capacity, countries that track their biodiversity across time. And to understand the differences more closely, I started looking at a case example from my own work. I've been studying the grasses of Madagascar for a number of years now, and we just published the book you see here. And we did an analysis to compare all the relevant statistics and discovery of Madagascar grass species versus British grass species. Madagascar is twice as big as the UK and it's got about twice as many grasses. Grasses are a fantastically useful model system because grass diversity is even across the world, and it works in similar ways in the tropics and in temperate regions. The most common British grass has been recorded almost 300,000 times, okay? The most common Malagasy grass has been recorded 300 times, so I was a bit shocked when I saw the statistic, even though I've worked in both areas. So this is a 3 orders of magnitude difference. British grasses, at least the most common one, has been recorded 1,000 times more often. Ouch. And this is my working process when I study the grasses of Madagascar. So I live in Britain and I work in Kew, which is marked in red, and I use the big tropical African reference herbarium in Kew. And then I have to go to Madagascar, of course, to collect plants, to work with local botanists, to work in the local herbarium, which isn't unfortunately sufficient. For the best herbarium collection of Malagasy plants, I have to go to Paris, which is in orange, in France. But then I have to also go via South Africa to look at their herbarium to understand and reconcile the species naming system with the South African grass species naming system. And I'd like to propose, in the time I have left, that foreign-driven science has fundamental consequences for how we understand biodiversity. No specimens were kept in Madagascar until 1948. They're here. This is a picture of the Jardin des Plantes in Paris. Remote taxonomy is inherently lower quality. This grass that you see on this slide was classified as 4 different species that look superficially different to anybody that hasn't seen them in the field. And this is all really important for our levels of understanding. Our worst understanding, possibly actually no proper work has been done outside the seed plants. So our paper didn't cover anything outside the seed plants. But some data is available on plantsoftheworldonline.org. There are 29 species records we haven't looked into. Examples of them are here. In case anybody wants to pursue that, I'm sure Raphael's very open to collaboration. And to finish off, I think I would like to conclude that clearly many more plants are extinct than we realize, looking at this pattern, many, many more. And the key driver for why our knowledge of extinction is so imperfect is that many parts of the world don't have the capacity and don't have the ability to do their own botany and plant recording. And foreigners coming in to do plant recording and botany, for example, in Madagascar, will never produce a quality of science and the quality of extinction records that we really need. The global knowledge is desperately uneven, and we need to understand the drivers of this knowledge inequality and work with poorer countries if we have any hope of understanding extinction. Thank you very much. Thank you. Goodbye.
And thank you, Maria. Our next speaker is Wesley Knapp, the Mountains Field Ecologist and Botanist of the North Carolina Natural Heritage Program. Wes will present Vascular Plant Extinction in the Continental United States and Canada.
Hello, everybody. My name is Wes Knapp. I'm a botanist with the North Carolina Natural Heritage Program. And I'd like to start out by thanking Rob and Larry and all the other symposium presenters for just making this such a wonderful event. Thank you for being here today. And I'm here to talk about vascular plant extinction in the continental United States and Canada. Most of this presentation will be based on a paper that has recently been published in Conservation Biology. This is an open access paper. It is readily available to the public. So if you're interested in what I'm presenting on today, though I can only kind of scratch the surface in this 15 minutes, please just check out this paper. It has a wonderful array of co-authors. A couple are part of the symposium, Reed Nosson, who's speaking next week, and Rob Noxie, our moderator. So a little background, I got very interested in extinct plants from an early professional age when I worked for the Maryland Natural Heritage Program, and my job was to discover rare plant populations. And the plant that captured my imagination was Nuttall's micranthmoides, because it is the only component of the Maryland flora that was ranked as GH globally historic. And I wanted to find this plant, learn everything I could about it, because I did not realize extinctions happened in places like Maryland. I thought it was places in the tropics or the Amazon. And I learned everything I could. And this is a picture, a slide, of an old herbarium specimen of micranthmoides. It is an intertidal mudflat specialist. I call it the opposite of charismatic megafauna, because it is a rather cryptic organism. You have to be at the right place at the right time to even find it. And it was last observed in southeastern Virginia on September 13th, 1941. It has never been seen since, and there are at least 3 duplicates of this rather famous location. And because I got interested in extinct plants, I talked to other botanists about the extinct plants in their states. And what I found is many botanists didn't know what extinct plants were found in their states. This was a real knowledge gap. And if you do some quick searches, you can find lots of information on lost animals, especially birds. The Hawaiian flora and plants are well known. And then extinction rates of freshwater fauna in this wonderful paper here as well. But the knowledge on botany and plants, or what was extinct in North America at least, was very poorly known. So the goal of this project was to compile a comprehensive list of the plant taxa believed to be extinct in the continental United States and Canada. We wanted to calculate extinction rates to set a baseline for the Anthropocene, because we'll see over and over again in the literature that extinction rates are expected to rise, but we don't know what the current rate really is. We wanted to draw attention to these extinct plants to drive future conservation efforts. What can we learn from these horrible losses, and how do we prevent future extinction events? So really the goals of this project. Now I have just a short bit of time, so I can't get into all the minutiae of the different grades of categories that can be represented with the umbrella of extinct. But the 2 most common categories are extinct or globally historic. The IUCN uses slightly different terminology for this. But this gets at the sliding scale of confidence we can express about something if it's truly gone, or maybe it's still out there somewhere in some suitable place. So I don't have time to get into that today, but what I want to say is that when I say extinct for this presentation, I really mean presumed extinct. Because extinction is a hypothesis that's very difficult to prove. You can't prove it, you can only disprove it by finding something in the field. And I hope every one of these extinction events I talked about will be found to be incorrect. They're all extant, but the data at this point does not lend itself to that being a possibility. All of these plants I'll talk about today have been searched for in their natural habitats that have not been found, some for hundreds of years. We also have another category we'll talk about today called extinct in the wild. We have a definition for that, which means no naturally occurring populations. And we're going to talk today about extinction since European settlement. Now, I'm not talking about extirpations because there are plenty of plants that are still found in Mexico that are lost from the United States. We are talking truly about globally extinct plant taxa. So the study area, it is an artificial study area. We're looking at the continental United States and Canada. And it is very similar, but not perfectly parallel to the Flora of North America project. Now, the Flora of North America project, which is a 28-volume product, which is almost complete, does include Greenland. And for this product, we did not look at Greenland. And the Flora of North America also looks at bryophytes in volume 27 and 28, and we did not look at bryophytes for this project. This is for the vascular plants. But I think it would be a wonderful addition to do the bryoflora, the extinct bryoflora of the United States and Canada. And the only moss I'm aware of is this Neomaculonia nitida, which is extinct from Canada. Now, we also did not want to look at Central America at this point because the flora is relatively poorly understood and a lot to be documented to found there still. We also excluded Hawaii and Puerto Rico because of issues of island biogeography. Methods are fairly rudimentary, lots and lots of literature review, the primary literature, floras, and manuals primarily. I talked to a lot of botanists across the United States, Europe, Asia, and Central America to get at the extinct plants from our study area. And lastly, we did a NatureServe Database Query looking at GH and GX plant taxa. Now, if you don't know what NatureServe is, it is an umbrella organization that compiles data from all these state natural heritage programs or Canadian province CDCs, Canadian data centers. And they upfill these data, take the data from the state level and upticks to the national level. And we run everything through an extinction matrix, which is what we see here on the left, with the most common plants being S5 or G5, moving up to G1 and S1. So if we say something is G1 globally critically imperiled, it comes with a sense of authority. But we have these other two ranks, GX extinct and GH, which means every state or province agreed that the plant was already gone, and we could vet that information further. But we had to come up with a way to vet these names in standard way, because as we all are aware, botanists don't always agree on what constitutes a valid species or taxon. So if we looked at just the NatureServe and the names from the literature, we had about 150 names, but not all names are created equal. So if we have Franklinia, for instance, it's the Franklinia tree, which is a very famous extinct in the wild plant, everyone recognizes that as a distinct entity. But then you have names like Rubus fryi. So that is either an extinct single-site endemic of West Virginia or it's not real. So there's two really extreme options on the table. So we had to come up with a method to vet all of our names under standard conditions. And to get to that, we developed the Index of Taxonomic Uncertainty. And this is the scientific consensus of a name. We think this is novel. This is something we developed for this paper. And I think it has some legs and some potential utility for other products, because most of the time people will point to a single treatment and say we're going to follow, nomenclature follows Flora of North America, for instance. But that's really just one or a few people's opinions on a name. This lets us look at the totality of the scientific literature to see what names should be included as legitimate or meritorious, rather. So we wanted to look at unique taxonomic treatments with different authorships. So we're not padding our data. And we didn't use products like the USDA Plants Database or BONAP or NatureServe, nothing against those products. We wanted to go to the primary literature where people had tried to apply these names in consistent fashions. So the Index of Taxonomic Uncertainty runs A to F like a grade school system. A is a unanimous recognition like Franklinia. No one lumps Franklinia. Category B, majority taxonomic acceptance. So accepted by more than 50% of the sources in the literature. This is something like Juncus purvitus, which is a single-site endemic to Massachusetts, very rarely considered away from Europe. Category C, this is minority taxonomic acceptance, accepted by less than 50% of the sources, but still numerous sources recognize this as a legit or meritorious entity. Then we had categories D's and F's. This is where we drew the line for this paper. You're getting to taxa that are extremely rarely recognized, or in the case with category F, they were described into science and then never recognized again. So for this paper, we looked at categories A, B, and C, which gave us 51 species or 65 total taxa that are believed to be extinct or globally historic from the flora range. This is a 108% increase from any previous study in the flora region. We have 42 taxa, 64%, which are global single-site endemics. Now, I want to put an asterisk next to this and say just because it was collected at one place at one time Doesn't necessarily mean it was a global single-site endemic. That might have just been the only place it was ever collected, but these are some of the limitations of the data. Now, only 2 of these 51 species and 65 taxa were listed as extinct by the IUCN, so there's a little disconnect between North American data and the IUCN data. And if you look at the total flora of the North US and Canada, which is about 16,000 species, and you divide it by our extinct plants, it's about 0.4% of the flora is extinct. And if you use a first contact date with Europeans in 1565, so we have 455 years, we're losing about 1.5 taxa a year, or 0.14 taxa a year, or 1.4 a decade. Now, the results at the diversity level are kind of surprising. 34 families, and some big families have big losses, like the daisy family, the beans, the roses, the borages, which are really diverse out west. But then big groups like the grasses, the orchids, and the sedges each only had 2, 1, and 1 respectively. We expect the grasses and sedges to be an under collection in a sampling bias, because botanists tend to ignore these entities. We had 53 genera. The biggest genera of loss were Crataegus, the hawthorns, which have seen apparent losses in the east, Astragalus, which is in the rose Fabaceae, and then most interestingly, Cryptantha and Plagiobothris, which represent all 6 of the Boraginaceae extinction events, and these are all located in one sub-tribe, so there could be some real phylogenetic consequences here. When you map out the extinction events, with the red dot being a specimen and these 2 blue dots being the broader, blue ranges being the broader ranging species, you'll see we have really heavy weight to the southwestern United States. This could be an artifact of biodiversity, but we think it also has something to do with botanical exploration for habitat destruction. The east was settled before it was ever inventoried, where at least the west had minimal exploration before habitat destruction by Euro-Americans. Not much in the upper Midwest, or in biodiversity hotspots like the Apalachicola or the Lake Wales Sand Ridge. We found that pretty interesting. These are probably just artifacts of data limitations and sampling. We have 4 species that have been described or recognized as distinct only from old herbarium specimens since 1995. There are still likely undescribed species in the herbarium record. Just this year, 2020, my colleagues and I recognized Marshallia grandiflora as a 2-county endemic in western North Carolina, and redescribed what's commonly attributed as Marshallia grandiflora now as Marshallia pulchra. So that's 100 years it took for someone to recognize that as an extinct entity. Now, this Govinia floridana here, which was described in 2000, was extinct most likely from overcollecting. You'd almost expect if a group was going to go extinct because of overcollecting, it would be orchids, but that's not always the case. We've recently learned that Digitaria filiformis variety Levaglunus, there are 28 herbarium sheets worth of material from this single-site endemic grass from New Hampshire. This most likely had something to do with the extinction of this species. The same can be said for Juncus purvitus, a single-site endemic to Massachusetts. There are 56 herbarium sheets. Almost all of them have copious amounts of rhizome rooting material like you see here. Now, extinct in the wild is a category that's very fascinating. These plants are no longer known from the wild, but known from botanical gardens or collections. We have 3 categories here: EW, extinct in the wild; EW question mark, which are plants we could not confirm the identities of, but we hope the gardens who are reporting to have this material will work with us to confirm this. Then EW star, which is something I have not personally seen, but a colleague had. But the most fascinating story of these EW plants is Arctostaphylos franciscana. This shows me kind of, I think this is what's going to happen in the future with many more plants. This was thought to be extinct before it was rediscovered in 2007. Unfortunately, it was right next to a bridge leading up to the Golden Gate Bridge, so they just picked it up and moved it. So by definition, it doesn't have any naturally occurring populations, but thankfully the species is no longer extinct. Now, when we talk about extinction, we like to know the causes. It's often very, very difficult to know why something goes extinct. Habitat destruction and alteration are by far the leading significant factors. 42 extinct species were single-site endemics, and the sites are now destroyed. But oftentimes it's difficult to know. Even Franklinia, the most famous of the extinct plants in the Americas, we don't know why it was gone. It was just collected one time by Bartram. And the other 2 species, Crataegus fecunda and Micranthomoides, which have broad ranges, we don't know what happened to these. We assume it's the death by 1,000 cuts, but it's often difficult to know. So part of this talk, I wanted to be a call to action. How do we learn from these extinction events? Because I'm not truly an academic scientist. I like to have things to impact the world in the field now to make our lives better. And this is no different. So we want to prevent extinction. Preventing extinction is the lowest bar we can set for conservation success, and we're not always successful. So given the results from the previous study, we need to prioritize single-site endemics for ex situ and in situ conservation. And this plant here, Packer serpenticola, is a great example of this. I'm working with Anne Francis and Amanda Trayer of NatureServe to identify global single-site endemics to try to get these into conservation gardens. Now this data is still very preliminary, but when you overlay, there's a map, NatureServe's map of biodiversity importance of just G1 plants, G2 plants, and Endangered Species Act listed plants. You'll see we have single-site endemics and biodiversity hotspots and a smattering of other locations. And we need to get these things into ex situ collections promptly. So how are we doing with these 89 plants? You'll see 9 of them are currently listed by the Federal Endangered Species Act. 38 are in the CPC National Collection, which is wonderful, and 55 in the BGCI collections, fantastic, but we need to do better to make sure these are all being captured. And I put a picture here of Solidago plumosa. This is a single-site global endemic, so it fits this category, and it was petitioned but decided not to be listed as federally endangered by the feds. Not disparaging that decision. I'm just making sure everyone knows that just because something is a single-site endemic, it does not mean it will be protected. So extinction is more common than we previously documented, 65 taxa total. Global single-site endemics need to prioritize conservation attention. Ex situ and in situ conservation is absolutely critical. Taxa are slipping through the cracks. We all need to work more collaboratively because the stakes are too high. Lots of people acknowledge, don't have time to do so. I look forward to the questions.
Thank you, Wes. Now we will have our live question, answer, and discussion with the speakers. I invite you again to submit questions using the Q&A feature of Zoom and the chat of YouTube. Please remember to direct your questions to one of the speakers, and feel free to give us a thumbs up if someone has asked a question that you like. At this time, I invite today's speakers to join us live for question, answer, and discussion. Okay, so fortunately, we have a number of questions that have been coming in, and I'd like to begin with Maria. Do language issues affect unequal knowledge about threatened plant species? For instance, are some plants being recorded in local languages that international researchers cannot access?
Of course, that's definitely the case. As a professional taxonomist, I'm very accustomed to using lengthy, incomprehensible Latin binomials. And I think that as a professional community, we all have a duty to make our information accessible to everybody, translate our names into common names, translate them into all the local languages. I think the bird community are doing a great example of data integration and communication, and we must build our plant sciences community to do similar work.
Thank you, Maria. This next question is for Wes, but I'll begin with it because it's directed to NYBG. Does NYBG have a list of plants that will be extinct in the next 10, 20, 50, 100 years due to climate change? Well, climate change and its effect on plant conservation is one of the active areas of research here at the garden. And we work very closely with a number of the conservation organizations throughout North America, especially the various state natural heritage programs. So we ourselves are doing a lot of the work, especially based on inventory and records from the past in our herbarium. But at this point, I'd like to turn to Wes and have him comment further on the actions of various conservation agencies, especially the state natural heritage programs.
Yeah, that's a great question. And I haven't seen any analysis of hard and fast predictions of what is expected to be extinct. I've seen some unpublished information on species that were likely to go extinct because of human population growth and sea level rise, for instance. But at the natural heritage program level, those type of threats are incorporated into the global rank. So anything that's G1 is more likely to go extinct than anything else that we track. But those G1s could and should be further prioritized with more detailed information on the readiness or speed at which they are expected to go extinct.
Could I comment on that, please, Rob?
Yes, by all means.
There are things that we can do. We know that species, particularly tropical species, are going to move, upslope as the climate warms. So species and habitats that are at the sort of the top end of a mountain system are going to be vulnerable because they're going to run out of space. We may have very few options there, but a greater worry is that species survive in highly fragmented landscapes. And so a species may be here and it wants to go there, but it can't go because there's a cattle pasture in between. And so re-establishing habitat connections is a very important and practical way in which we can address the problems of climate disruption. And taking the opportunity to promote what I do, I direct an NGO called Saving Nature. And what we do at Saving Nature is work with local conservation groups to build habitat corridors of native vegetation. So species are going to move. We have to give them the freedom to move. And that means ecological restoration. The UN has declared this decade the decade of restoration. I think there are very practical things we can do, and we need to be doing them.
If I could just chime in on that. Stuart's point about putting natural connections in the landscape is just absolutely fundamental. So we might even think about not just a decade of restoration, but a decade of reconnection.
Yeah.
Thank you, Tom. And this next question is for you, and it's even more fundamental, but it relates to a point you made in your introduction. Tom, what are your best strategies for alleviating plant blindness and thus providing some hope for alleviating the immediate crisis of plant extinctions?
So I think the basic answer to that is get more people outdoors and looking at plants, not just the animals and habitats. And one of the things I was thinking about as all these great presentations were going on is the incredible data sets we have for birds from amateur bird watchers through the eBird program. And there's no reason at all that we can't have something similar for plants. And indeed, I mean, there are first steps in that direction in software like LeafSnap. But I think it needs to be thought about really on a very aggressive and strategic kind of way.
Thank you, Tom. And I'd like to follow up with Maria on much the same question. One of the questions that's posed right now is, Maria, do you think countries of the global south have enough taxonomic expertise to document plant extinction? I don't see the young generation or even our university system paying serious attention to teach plant taxonomy. We are in a serious stage of plant blindness, even funding agencies paying more attention to animal conservation than plant conservation. So what is your take on this?
It's an incredibly important question for all of us to think about. And I'm pleased to say that I have worked with many students in Madagascar, in Kenya, in Tanzania, in South Africa, in Colombia. And there's an incredible amount of interest and commitment from young people and from professionals across the global south, across poorer countries. It's up to us, however, to help with resources. And given some support, I think any country is capable of growing its professional expertise and really recording and looking after its biodiversity.
Thanks, Maria. Okay, so Stuart, how about you? And others certainly please chime in. Can GMOs, genetically modified organisms, play a role in bringing back extinct species?
I am a critic of de-extinction. And I am for the following argument. It creates what I call a moral hazard. I've testified many times in front of committees in the House and the Senate And there's always those who say, you know, it's okay if we destroy the world's forests, it's okay if we destroy the Pacific Northwest. You know, if we drive a species to extinction, we can always bring it back from animals in captivity, or we can always, you know, re-engineer the species and put it back like the passenger pigeon. Those techniques are clever and they seem very sexy because they're so high tech, you know, visions of Jurassic Park. But of course, what they do is they allow people to, you know, destroy the natural world that we have and think it's going to be okay because technology can fix things. So I think we have to be very careful about going down that route. We need to protect the species that we've got. We need to stop deforestation. We need to do something about global change. We need to remove invasive species where they do not belong. I could go down a long list. Those are the things that we have to do first.
Wes, this question's for you. If XC2 conservation of single-site endemics is of such high priority, would it not be a good idea to build a global facility with branches in several countries where single-site endemics are cultivated to prevent extinctions?
Absolutely, that's a great idea. I wish I had the funding to do something like that. But I know we have large XC2 conservation efforts globally, namely seed banks. And those are really critical, and they can be a last line of defense for preventing extinction. Now I'm working with a number of conservation gardens here in the US to identify and grow some of these last living individuals, because the seed banks aren't necessarily all we need, because we also have to have the knowledge to grow these plants after we've collected the seeds if we need to germinate them again. But I think that is an absolutely wonderful idea.
So here's another question about single-site endemics. Do you think there are ways of predicting which regions host still undiscovered single-site endemics and thus give priority for preventing future plant extinctions?
Oh, absolutely. This could be done at a global or national level. Like in the United States, most species that are being described are in biodiversity hotspots like California, the Southwest, and the Southeastern United States. Those would be relatively easy to identify, but at a small scale, it's actually surprisingly difficult. Because as you saw on my map very briefly, we have single-site endemics in places you may not expect them, like in the Northeastern United States and the Northwestern United States. So it can't be perfectly predicted, but I think that could help.
Okay, thank you, Wes. Tom, what are your thoughts on assisted migration for plants that are at high risk of extinction in their native range due to climate change, habitat destruction, and other threats?
So in a sense, what I was talking about before, but about building on Stuart's comments about putting connections back in landscapes is a form of assisted migration, as opposed to actually planting some particular endangered species where you think its preferred climate will actually occur. I can see some of that as part of what we need to do, but the most important thing is to take on climate change in a really serious way, which we are not globally, and energize a younger generation in the fascination of the natural world and plants in particular. Maybe create something in this country like the Civilian Conservation Corps, which could be involved in this kind of activity, and even make that a reasonable element in what the Peace Corps does. But we just really need a lot of young people to rise up and think about how they can make a better world for future generations.
Thanks, Tom. This question's for everyone, and it relates back to the seed bank matter. So are there any extinct plants that are currently in seed banks? And if so, do you know any actual examples, and might they be used to bring back extinct species?
I'll start out. I know I had to contact numerous seed banks who reported having seeds of putatively extinct taxa, but we worked carefully to vet the identifications, and every one of those turned out to be an incorrect identification. Because if they were correct, it would have been absolutely critical to attempt to regrow these species. I mean, that is the intent of having those seeds in storage. But at this time, I know of zero.
Does anyone else know any examples, please?
Apologies if my question doesn't, if my answer doesn't directly answer the question, but I'd like to point out much following what Stuart said earlier. Having collections of seed does not guarantee, unfortunately, our ability to regrow the plants. Plants are often associated with specific mycorrhizal fungi. Plants often have exact relationships with insects. Many plants cannot be grown from seed, and we simply have no idea how to cultivate them. More than half of global plant species diversity has never been successfully cultivated at all. So by far the easiest solution is just to not let it go extinct.
Amen.
Maria, while we have you, we have some questions about building capacity. So here's the first one. Is there an international effort to support the establishment and operation of herbaria in countries that lack the resources on their own to support herbaria?
Rob, of course, the best international effort to coordinate herbaria globally and support them is your one at the New York Botanical Gardens, and it's called the Index Herbariorum. Index Herbariorum coordinates global information on all herbaria worldwide, and I believe it provides some resources. Of course, financial support does remain a very difficult issue.
So following up on that, Maria, what would you recommend? Obviously, financial support is a major problem, but what are other strategies you recommend for building capacity in parts of the world where it's most needed?
I do have one suggested action for everybody listening to this today. I suggest that all of us download a copy of iNaturalist and all of us record plants growing near our houses, and all of us encourage our families and friends and our students to notice the plants that we walk past every day. And the growth in our capacity to appreciate, understand, and record plants does tend to also help grow funding and institutional support.
I'm very proud to be the godfather of iNaturalist. The scientific ideas were ones that Scott, Larry, and I discussed when he was a graduate student of mine. So, you know, it took Scott, I don't know what it was, a year or two to get the first 1 million observations, 10 months to get the second 1 million observations. They are now coming in at the rate of more than 1 million observations a month. It's a fantastic program. It's so easy to use. And it was designed very much so that we could bring plants and other taxa up to the level of eBird, which Tom mentioned. eBird is now closing in on 1 billion observations of birds. But you know, iNaturalist is catching up fast. It's a wonderful program.
Thank you, Stuart. Now,
oh, so I need to answer something that's just come in.
Okay, go ahead.
David Kuntz has just said not to post rare plants as they could be poached. Absolutely, but there are protocols within iNaturalist that we built into it at the very, very beginning that allow the person who submits the record to fudge where the location is, so that people can't go to that exact location to find it. So there are protocols for endangered species.
Well, we all know that we have to confront hard realities when dealing with conservation, especially the matter of extinction. And this next question really draws us to that point. So I offer it for all of you speakers, and I'd love to hear from all of you. And I think our audience would too. I'll read the question now. How can conservation of plant species be achieved in low-income countries? Like, I'm sorry, my questions bounced around. Here it is. How can conservation of plant species be achieved in low-income countries such as Zambia, where I live, where use and conversion of the natural landscape can mean avoiding destitution for rural inhabitants? Zambia will be defaulting on its international loans very soon, and I fear that this will result in increased woodland and grassland destruction or degradation as people rely more on the natural resources to ensure that they can survive. So I'd really like if everyone could comment on this very real matter.
So I think there is actually tremendous opportunity in all of that. Back in 1984, I proposed the idea of debt for nature swaps. And there was absolutely no reason that there cannot be debt for science swaps, ongoing debt swaps for habitat restoration, training of international capacity, a lot. And I know right now that even some large corporations, which are experiencing a lot of indebtedness, are also looking at that as something that they may be able to convert into something useful.
Tom, are you able to tell us maybe a quick success story about such debt for nature schemes?
Well, I mean, it literally went into the many billions of dollars. And you know, there are just a whole series of protected areas around tropics in particular that were done that way. And since we're celebrating Madagascar here appropriately with Maria, one of those was with Madagascar, and it actually started with USAID money, and then they used that money to buy Malagasy debt. And I cannot give you the multiple of what it did to the total amount of money they had as a result. So there's a lot of opportunity space here.
I mean, one of the tragedies of a country like Zambia, which I know reasonably well from the work that we do on elephants and big cats there, is people are moving into dry woodlands, into Miombo woodlands that have, you know, terribly poor soil. So the agriculture there is really desperate. And the challenge is for us to work with our Zambian colleagues to try and ensure that Zambia's wonderful national parks and even its hunting areas bring in income to the local communities. Those places are going to be so much better off if we can keep them in national parks and converting them to really quite wretched agriculture. But that has to make a difference to the local people. It has to work at the local level. And I think that's the challenge. We have to find ways of not destroying areas that are never going to be very good for people as agriculture, but could benefit them a lot more, recognizing that people want to visit and see the wildlife in such a wonderful place.
Any other comments on that one?
I would just like to add that as a global community, I think we have a responsibility for giving agency and giving power to local communities and to local scientists, because these local issues cannot be understood by us from New York, from London. They can only be understood at the local level. And the one serious problem at the moment is that often funding is effectively only available to foreigners, and we need to change that.
Okay. Wes, there was a question for you about California as being such a hot spot for plant diversity, but also extinction. Do you have any success stories from your colleagues in California about plants that have been conserved and saved from the brink of extinction there?
Oh, California is an amazing, amazing opportunity for exciting. It's so exciting, all the work they've done there. Dan Glusenkamp, California Native Plant Society, was one of our co-authors. He's the one who discovered that Arctostaphylos franciscana and helped ensure it didn't go extinct. The Center for Plant Conservation is stationed in California, and the work they do on seed conservation is absolutely incredible, because you're talking about, as someone who lives in the southeastern United States, which we think of as very botanically diverse, California is truly a level above that. And they have so many G1 narrow endemic plant species that if it wasn't for the efforts and money and time that they have spent in the understanding of the Californian flora, the extinction rates in California would be much higher than have already been documented.
Thank you, Wes. This next question is again for everyone. At what time and how do we involve the general public in understanding and collaborating to prevent extinction of plants? Are kids ready to be involved in this challenge, or do they have to wait until adulthood?
If I may jump in,
Please.
the question assumes that our work is separate from that of the general public, and our work should never be separate. It's for everybody.
So I would say that any city with an important botanical garden should actually make sure that all their school children learn about botany and conservation and sustainability.
And I'd like to just quickly chime in that anybody who's working in the overarching field of conservation, you're essentially preventing extinction. You're conserving the resource. And I'd also like to point out that voting is critically important in representative democracy, but every time we spend our dollars, those are votes. So if you're spending your money on sustainable products, by definition, sustainability will not promote extinction. So please make those informed decisions, because they have an impact.
So really this segues right into my final question for today, and that is, what can individuals do? You've partly answered this, but I hope that you have some more suggestions. What can an individual person do to help combat the plant extinction crisis?
Get out into nature.
Straightforward, right? I mean, you learn about it. You talk to people who are already engaged in it, find ways that you can bring others into the exercise. Knowledge and caring. Stuart, I'm sorry, I jumped on you.
Yeah, go ahead, Stuart.
Get out into nature, use iNaturalist, become informed, as Thomas said, and become politically active. There's no reason, we live in a democracy, there's no reason why one shouldn't go and visit one's congressman, one's senator, one's member of parliament, whatever country you're in, and tell them that you care about the environment. Take your children, take your students. The reason why bad things happen is that we often fail to engage the politicians who hear a different viewpoint from other people. There is no reason why we shouldn't be more active as informed citizens.
Any other comments? Well, we've certainly had a lively discussion, and I really wish we could continue. Before we wrap up for today, though, I remind you that next Tuesday we will resume this symposium with presentations from 4 more experts, followed by discussion. So please remember to register in order to join us. Also, please post about the symposium on your social media and draw others into it so that we can have a wonderful concluding session next week. Come prepared with questions and plan for more stimulating discussion. On behalf of all of us at New York Botanical Garden, I thank My Good Fund Trust and the Andrew W. Mellon Foundation for their financial support for this symposium. Also, we are most grateful to Wes Knapp and Douglas Daly for their assistance with symposium planning. In addition, for their thoughtful contribution, I thank today's speakers: Tom Lovejoy, Stuart Pym, Maria Vorontsova, and Wesley Knapp. Thank you too for joining today's first part of Here Today, Gone Forever. We greatly appreciate your participation. If you missed any part of this event or wish to watch it again, a recording will be saved and shared online via the NYBG lecture library. Once published, we'll send a link to everyone who registered for today. Today's speakers join Larry and me in urging you to engage in inclusive, science-based, and forward-thinking endeavors to minimize plant extinctions. Next week's speakers will provide even more recommendations. May you have great success with this important work. In the meantime, we wish you the best of health. Goodbye, everyone.
Bye-bye.